Part 1. Introduction.
Hi, my name is Rafael Erlich. I'm a postdoc at Professor Steven Rehen's lab at Federal University of Rio de Janeiro in Brazil.
Hi, my name is Ismael. I am a research assistant also at Professor Steven's lab.
In this video, we will show how to prepare cryosections of embryoid bodies. So, let's get started.
Part 2. Materials and equipment. For this procedure, we will need an embedding medium for frozen specimens, 4% paraformaldehyde solution for fixation of the cells, sucrose solutions for cryoprotection, a needle with a slightly bent tip, a stereo microscope for better visualization of the EBs, and a shaker. Part 3. Here, the human embryoid bodies are being cultured in non-adherent culture plates. Using a pipette, we group all the embryoid bodies as close together as possible in order to transfer them to a 15 milliliter conical tube. The EBs are carefully harvested and transferred to the tube. It is important to collect all EBs since there isn't a large amount of material. After EBs are decanted, the culture media is carefully discarded. You should be able to see the pellet at the bottom of the tube. Now we can proceed to the fixation procedure. EBs will be fixed in 4% paraformaldehyde solution in PBS, or phosphate buffered saline. It is important to resuspend the pellet with PFA solution.